Wonderful. Well, welcome. My name is Jorge Contreras. I'm a professor at the College of Law here at the University of Utah, and I'd like to welcome you to the Frontiers in Precision Medicine 3 conference. Will personalized medicine improve population health? So this is the third in our series of interdisciplinary conferences on precision medicine and personalized health. We began the series in 2015. Many of you in the audience were here with us at that time, so welcome back. As you may remember, the first event that we had in 2015, the topic of exploring science and policy boundaries in precision medicine. The next year, our focus was on cancer, big data, and the public. And this year, we've shifted to ask the very difficult question of whether the tools of precision medicine can affect a broad segment of the population and improve overall health. So this event brings together contributions from a number of units and departments across the University of Utah campus. Named up here on the slide, the University of Utah School of Medicine, especially its program in personalized health, the S.J. Quinney College of Law and its Center for Law and Biomedical Sciences, the Huntsman Cancer Institute, and our new Department of Population Health Sciences, which we'll hear a lot more about throughout the day. In addition to these internal groups, we've assembled a group of nationally renowned authorities who will work with us and engage in this discussion of this topic. So with that, I'd like to turn over the podium to my colleague, Dr. Will Deer, who's the Executive Director of our program in personalized health. Thank you.
Thank you, George, and good morning, ladies and gentlemen. In a December 2016 interview, Dr. Francis Collins, director of the NIH, predicted that, and I quote, "Precision medicine is going to change everything about how we understand health and disease." The Precision Medicine Initiative, announced by President Obama in 2015 and now renamed the All of Us Research Program, plans to study a cohort of at least 1 million Americans. It seeks to expand our understanding of heredity and disease, revolutionize the treatment of disease, and improve human health. And according to the White House, precision medicine is, and I quote, "health care tailored for you." At its most fundamental level, precision medicine seeks to optimize treatments based on individual physiologic characteristics, such as the ability to metabolize certain drugs, or response to one or another set of interventions based on individual differences in genes, environments, and lifestyles. Now, during the same time, national experts have highlighted the importance of research in population health, which rests on the belief that improved life expectancy, better quality of life, and health are all shaped by interdependent social, economic, environmental, genetic, behavioral, and healthcare factors. As such, population health science is an interdisciplinary field involving cross-sector collaborations to address complex population health problems with multifaceted interventions. And we are fortunate here at the University of Utah to have one of the few population health sciences departments led by Dr. Angie Fagerlin, who will be moderating our first session. Now, with the importance of both precision medicine and population health sciences, numerous questions have been raised by a variety of experts, such as, will the costs of specialized medicines and diagnostic tests in cost-constrained environment be affordable? Will resources utilized from such specialization and targeting of individuals negatively impact resource needs, such as vaccinations and other preventive interventions which improve a broader populace's health? Will there be appropriate attention and focus to improving outcomes in all of our community members, not just those who can afford to pay or who have access to our healthcare delivery system? And will we sufficiently assess the needs, wishes, and desires of our fellow citizens in our communities with interventions that are being considered or being developed? Thoughtfully applying these approaches in a cost-constrained environment is one of the great challenges for stakeholders in the healthcare arena. There is inherently greater simplicity in a single approach to improving healthcare. For example, there's a body of research suggesting that there is little correlation between higher spending and higher quality healthcare, and it has helped popularize the perception that less care is better care, or even that more care is harmful. But as Dr. Lisa Rosenbaum has recently written in the New England Journal of Medicine, and I quote, "Though reducing unnecessary care is important, the missionary zeal of waste hunting begins to obscure a complex evidence base." A complex evidence base. And the evidence base of one or several clinical outcome studies consistently supporting the use of intervention A versus the use of intervention B obscures the reality of a subset of non-responders to the better therapy intervention A, or potential safety issues in a subset of individuals who receive intervention A. Thus, the notion of a one-size-fits-all solution to all complex chronic disorders is also misguided. The complexity of human biology demands that we employ the breadth of our capabilities and insights to personalize our medical interventions where necessary and where possible, while ensuring that resources are used to maximally benefit the broader population. Dr. Contreras and I appreciate your attendance and participation in this symposium. I'm pleased now to ask Dr. Angie Fagerlin to the podium to lead our first session. Angie?
Thanks. And I'm just so excited to get to this, so I'm going to do a very brief introduction. I'm thrilled to present the co-opening keynote session, Will Personalized Medicine Improve Population Health? And our first speaker will be Dr. Maureen Curry, who is the founding director of the CDC's Office of Public Health Genomics, which was formed in 1997 to assess the impact of advances in human genetics and the Human Genome Project on public health and disease prevention. In this work, Dr. Curry leads national efforts for integrating genomics into public health research and programs for disease prevention and health promotion. Dr. Curry has over 500 scientific publications, including articles, books, and book chapters. Since 2007, he has served the National Cancer Institute as Senior Advisor in Population Health Genomics in the Division of Cancer Control and Population Sciences. He has led the Epidemiology and Genomics Research Program at the NIH. Dr. Curry has won numerous awards for his science and leadership, including the CDC Research Honor Award for Outstanding National Leadership in Genetics and Population Health in 2000, and the National Cancer Institute's Visiting Scholar Award for Leadership in Vision in Genetic Epidemiology and Public Health in 2005. And we're so excited to hear what he has to tell us. Thank you.
Good morning, everyone. Can you hear me? So I'm delighted to be here, part of this conversation today, and you stole my thunder earlier, but just wanted to first thank the organizers for doing this and tell you my story. So the caption here is the elephant in the room, and that's sort of what precision medicine represents for some of us in the world of public health. Because as our opening speaker mentioned, the issue of resource needs always is in the background picture. Will more healthcare lead to better population health? I've revealed everything on the first slide. So the answer to the question, will personalized medicine improve population health, is yes, if. And I'm going to lay out 4 themes to tell you what the if is or are. Also, some of you may be interested in watching a debate I had 2 years ago with the Dean of a School of Public Health, Sandro Gallia from Boston University. And we had a point-counterpoint where his answer to the question was no, and my answer was yes. So I'm going to try to channel Sandro Galea this morning, but I believe in the yes, but there is an if to that. Last but not least, the word personalized medicine to me is an oxymoron. All medicine by definition is personal. So if there is a way to do it better with more precision, that spills over to the population level side, which means we can do public health with more precision. So the word precision is my preferred word, but we can debate that later. So that's the article I wrote up with Sandro Galea in JAMA a couple of years ago. And by the way, if you open up JAMA today, there is another piece that asks the same question. Just found it this morning from that came out from the workshop of the National Academy of Medicine workshop last November. So the first theme is very obvious, and I'm going to fly over 30,000 foot level in most of the conversation this morning. So we know that disease is complex, and nature and nurture work together in mysterious ways. So nothing is genetic, nothing is environmental. There is a complex web of causation. And my favorite article was from Nature a couple of years ago, in which investigators looked at the heritability of dozens and dozens of health-related traits. And they found out that 50% of these traits are genetic and 50% are environmental. I don't think that advances the discussion by a bit. That's why I don't like the concept of heritability, because it masks underlying interactions between genes and environment. I like to think about disease processes as a stew, not a pie, because the pie you always divide it into either, is it genetic or is it environmental? Some of the genes, even for genetic disorders, need environmental components and vice versa. So simplistically, we divide diseases into whether they're genetic or non-genetic. The genetic disease milieu is more than 7,000 diseases, and if we hear our next speaker, she'll tell us there is more hidden in plain sight. That's the next talk. But most diseases are complex in nature. They involve genes and environments and social and environmental factors. And the genomes now are interacting in very different ways. I mean, only data will tell us how they will interact. We're more microbes than human cells and genomes. The cancer genomes evolve and interact, and they lead to different outcomes. And then there is the genomes of other organisms, vectors that transmit disease. So the world of genomics is really moving very fast. And on top of this, the whole area of epigenetics is promising for us to shed a new light on the concept of gene-environment interaction. For the first time, we are able to measure the impact of environmental influences on gene expression and whether gene expression can be inherited throughout generations. This is the famous agouti mouse. These are two identical genetically identical mice. One of them has the turned on the agouti gene through methylation, which leads to a metabolic syndrome. So studying epigenetics is promising a new era of gene-environment interaction. So why am I introducing this at this point? Is because every health problem in the population needs to be approached from both an individual and a population perspective. This is a paper from our previous CDC director, Tom Frieden. And he talks about the health impact pyramid. And these are examples throughout. But at the base of pyramid is fixing socioeconomic factors: poverty, education, housing inequality. There is nothing personalized about this. I mean, this is the base by which you can make the largest impact on population health. And as you go up to the top of the pyramid, and like counseling and education and even healthcare interventions, yes, you're going to be making an impact on individuals one at a time, but their impact at the population level will be less. But make no mistake, every problem, whether it's hypertension, whether it's cancer, whether it's smoking cessation, will require all of these parameters working together. And the question for me has been all along in a public health environment is, can we do all of these things with more precision, leading up to the healthcare interventions that are so-called now personalized? And So the debate in public health goes something as follows. And Sandro, I mean, the reason I invited Sandro to the debate, because I read his New England Journal of Medicine article with Ron Bayer, who essentially had not a very favorable impression of public health in the precision medicine era. And I quote just one of the many sentences in that article. "We worry that an unstinting focus on precision medicine is a mistake and a distraction from the goal of producing a healthier population. The more we spend on healthcare, the less we spend on other things that we know work." And that's sort of the tenet of the conversation. At that same time, when that paper appeared, Ron Zimmern from the UK and I wrote this blog saying precision medicine versus public health, it's a false dichotomy. We should stop pitting the world of public health against world of medicine for two reasons. One, public health always loses, but B, I think we have a chance to work with the biomedical enterprise to improve health at both the individual and the population level, and here's how. So my second theme is, as medicine becomes more precise or personalized, depending on what word you want to use, we need public health to ensure its population health benefits. In other words, a population is more than just a set of individuals coupled together. There is a population level structure and capacity and infrastructure that is needed for implementation. And I laid that out when the Precision Medicine Initiative was announced in a JAMA article with my friend and colleague from UNC Chapel Hill, Jim Evans. And it's laid out as the public health perspective on the National Precision Medicine Cohort, balancing long-term knowledge generation with early health benefit. So that sort of goes without saying, but today, the genomic landscape vis-a-vis testing is growing very rapidly. And a place to capture that evolving knowledge is the genetic testing registry that's maintained by NCBI. And the last time I checked, at the end of January, there were 54,000 genetic tests to more than 10,000 conditions with about 16,000 or more genes in more than 500 labs. So that alone, and these numbers are going up, and they are applied throughout the whole disease spectrum continuum, from risk assessment to prevention, to treatment, to optimization, to screening, and more to the direct-to-consumer market. And you'll be hearing a little bit more about that later on. But people want this information. People don't know how to use this information. There is a lot of discussion about the validity and utility. And now with whole genome sequencing as a platform, or whole exome, the need for doing test by test or gene by gene analysis may be overcome by this. So what is the role of public health to ensure the population health benefits of these advances? So the first one, which is I think very crucial, because that's where we need to find out what works and what doesn't work, so that we don't harm people or we don't use healthcare resources unnecessarily, which is the identification of genomic applications that are supported by evidence. And yes, evidence matters, data matters, and we need information on both the validity of these applications as well as the balance of benefits and harms. More importantly, the population health, both applied public health and the scientific arm of population health, should be able to lend its collaborative lens to the discussion of how to quantify the burden of preventable disease, assess the impact of interventions at the population level, not just in small studies, but what would happen if these things are rolled out at the population, modeling healthcare costs and saving, barriers and facilitators and ethical issues, and addressing health disparities in a major way. And then last but not least, lab practice, because not all lab tests are created equally, and sometimes we take that for granted. The same assay can give you different result by different labs. So to cut the long story short, we at CDC in our office has been working on evidence, and I was glad to see Jeff Botkin here in the audience, who was part of this discussion we've had over years when we set up a special panel called EGAP, the Evaluation of Genomic Applications in Practice and Prevention. I'm not going to talk about that today because there is no time, but I wanted to tell you where we end up today. And people and programs at CDC and healthcare always ask me, so what is ready, what's not? So we wrote this paper a couple of years ago, and we tried to keep it simple. Tier 1, Tier 2, Tier 3. And some of these things are being used now. I'll show you a slide later on. But Tier 1 are those genomic applications that are supported by evidence, for which there is an evidence-based recommendation by a group. Newborn screening is the ultimate public health genetics program. I mean, forget about the new tools. We've had those applications for more than 50 years now, but there is more. Tier 3, on the other hand, are those where we don't know, or there is enough evidence that they don't work or they do more harm. I lumped the direct-to-consumer market here in that Tier 3. Some people may disagree, but I just don't see the level of evidence that's needed to use direct-to-consumer genetic tests in the population. Tier 2 is where the action is in between. A lot of pharmacogenetic tests are there because you have robust associations, but we don't know ultimately whether or not if pharmacogenetic tests, or many of them, are used in clinical practice, whether the benefits will outweigh the harm, or that's the clinical utility. So believe it or not, there are lots of Tier 1 applications, and we try to maintain in our public health genomics base online what's new, what's not. And last time I checked, there were 68 Tier 1 genetic tests. More than half are cancer. That's why I spend a lot of my time in the cancer program at NCI. There are more than 100 Tier 2 tests. Many of them are pharmacogenomics. So if you search that database, you'll see information on guidelines and programs, and you'll see what the information is supposed to be used for. So to me, that's a starting point for the discussion, but that's an evolving field. Every day we have papers that come out that evaluate the validity and utility. So in the last 2 to 3 years now, we focused on a few top Tier 1 genomic conditions for public health purposes. I'm going to describe 2 cancers and 1 heart disease. BRCA-related hereditary breast ovarian cancer and Lynch syndrome, and also familial hypercholesterolemia. Collectively, these conditions affect more than 2 million people in the US. Most of these people don't even know they have it. Implementation of evidence guidelines will actually save thousands of lives every year. And so we've been working with health departments, Utah is included with them, and others to develop toolkits for public health departments, and working with the CDC programs and external partners, primarily cancer and heart disease. So these are the 2 genetic conditions affecting breast, ovarian, and colorectal cancer. Of course, you all know the actress, Angelina on top, but you may not know Georgia Hurst, who's a Lynch syndrome patient, and her son. She's has been an advocate for the community, trying to inform people about their options. So there are 2 evidence-based recommendations, one from the task force and one from our own EGAP working group, that if implemented, can actually save lives of these people. But notice the wording of these recommendations. One of them is screening to identify family history associated with BRCA, using genetic counseling, and then BRCA testing when appropriate. With the EGAP, it's more cascading, testing all colorectal cancer, and then cascading to the relatives. So for those people in public health, they say, well, but if you implement all these recommendations, you're only going to get at a slice of colorectal cancer, only a slice of breast and ovarian cancer. And they're correct. But these thousands of people, or the 2 million people that are served, I mean, this dwarfs the newborn screening program by leaps and bounds. I mean, we screen 4 million babies every year to find 400 kids with PKU. Think about that for a minute. I mean, there is an opportunity to affect population health in the millions if an adult genetic screening program can be studied, evaluated, and all the bells and whistles worked out. So CDC has been funding some of the cancer programs, and the current funding from the cancer division, it does involve Utah and Colorado, Connecticut, Michigan, and Oregon. Earlier in the mid-2000s, we funded 5 states. Utah was one of them. But here are some of the program activities that are mentioned. I don't have time to go through this, but the idea of getting public health departments working with academia and the healthcare system to educate them, to implement surveillance at the population level, policy or systems change is an idea whose time has come. And I think we need more of it rather than less of it. And so just by the way of an example, Michigan has been one of the states that have embraced genetics a long, long time ago. And through both their two strategic plans, one, their genetic strategic plan and the cancer plan, they've integrated genomics into their way of thinking. And over time, they've been able to, let's say in the case of BRCA, more cases found, more counseled people. This is statewide programs, data presented by Deb Duquette, who used to be the state genetics coordinator in Michigan, recently left the state to pursue academics somewhere else. Now, one of the reasons I always focus on the population level is because of disparities. Disparities are with us whether we like it or not. It involves disparities in discovery, but disparities in implementation. In this case, here are two data points showing the disparities in BRCA testing by race and ethnicity and within rural-urban differences. The panel on the right is our own paper published last September in the MMWR showing with large data, administrative data, the trends in BRCA testing in the US based on millions of records and the differences between metropolitan and non-metropolitan area. Notice the upswing after 2012, which may be due to many factors, including the Affordable Care Act, including the Supreme Court Myriad decision, or the Angelina Jolie effect, if you will. But there is always be a difference between rural and urban areas in implementation. Race and ethnicity goes without saying. We have data from multiple disease endpoints to show that BRCA testing in young women with breast cancer is underutilized by race and ethnic groups. And we need to do better at the population level in order to fulfill the benefits of precision medicine for all. And I can't come to Salt Lake without mentioning my friend and colleague, Roger Williams, who was the initial founder and pioneer in familial hypercholesterolemia. Roger, I've known, and his untimely death in 1998 really put a damper on the field, but the field is now picking up steam through the efforts of many, many groups, including Catherine Weileman, who is the president of the FH Foundation, and Catherine, who is a patient who had her first heart attack at the age of 37, has taken it upon herself to gather this team and push the global FH agenda forward. So Utah has a long and distinguished track record in FH. And I don't have time to go through this, but the MedPed program has been the template with which current efforts are being organized. But the data that is emerging now, it's even more fascinating. It's more common than we usually thought. It's 1 in 250 disease, 1 in 250. Imagine, show me another genetic disorder that's that common. And there are evidence-based recommendations that works. We need aggressive treatment, and we need to do cascading in relatives. It's highly underdiagnosed, highly undertreated. There are racial ethnic disparities. Therefore, missed opportunities for public health and healthcare to partner together. I just wanted to show you our own data, which will be presented next week at the American Heart Association and published later on. This is data from NHANES. NHANES is a national health examination survey, which is a nationally representative sample of the US population. Cut the long story short, there is no good way of finding FH in NHANES because you have different diagnostic criteria, but we estimated from NHANES that it's 1 in 250. And we looked at utilization rates, so there are 3 sets of bars: the general population, people with high lipids, and then the putative, or we think that these are FH. And people, and even with FH, that's the bar on the left-hand side, are not, half of them are not even taking statins. And a lot of them are not taking high-intensity statins. So therefore, we know, and we, for the first time using population-based data, we show that there are undocumented cases of FH and opportunities for treatment. And we have further data that shows young and uninsured patients within NHANES are at the highest risk of undertreatment. So nothing new here, but it's important. So the third theme I would like to elaborate on is that as implementation goes forward with the tier ones, the public health sciences have a lot of role to play in carrying over that tier 2 and tier 3 over the hump, or at least finding the nuggets in those to develop and implement new knowledge. What do I mean by that? So we're all excited about the bench to bedside. And a few years ago when NHGRI released their new strategic plan, which was back in 2011, they're going through another cycle right now for strategic planning, everybody declared that the future is now. And what they mean by the future is now that we've arrived to the bedside. But arriving to the bedside is only the beginning of a long journey. And what I mean by that is that we need to do all these other steps that show, once you get to the bedside, evaluate to get to evidence, and from evidence, do the implementation science and the health services research to integrate into program. And then from there, evaluating what needs to be done at the population level. And we've been looking at this time after time. We've published so many papers on this. It seems so repetitive now because we always find the road less traveled. When we look at all the published scientific articles in genetics, we find that 98 to 99% are either discovery or early translation. Nothing, the 1 to 2% is in that whole realm of public health sciences, clinical and population sciences. We need to invest in the road less traveled even more in order to fulfill the promise of precision medicine for the population. So there are a few health systems that have begun integrating genomics into their midst. And I could have used examples from Utah, but here I'm using Geisinger as an example from Pennsylvania. And my code has been started many years ago. My code is based on whole exome sequencing of anybody that walks through the door. And based on the return of results from the ACMG list and more, they're beginning to find people in the population of Geisinger that otherwise have not been found before. So this is the last time I looked at their data. They have a fresh batch in March. So right now they've returned results to more than 500 people. And guess what? Most of them are in our top tier 3, the 2 cancers and familial hypercholesterolemia. They published a number of papers that show that these people were not essentially being diagnosed if not for whole exome sequencing in the population. And there's a bunch of other things. And the panel on the left shows you from a recent paper we published that in order to do this, you need more implementation science. So when we looked at the literature and implementation science, we found it a little bit lacking. So we had the politically correct title of opportunities for improvement, but you have to read the paper to see what's going on. I would love for pharmacogenomics to succeed because pharmacogenomics will affect everyone in this room and beyond. And so many of us are on so many medications. But the reason I put it in tier 2 is because there are pockets of success but not ready for large-scale implementation. A couple of examples, and I'm hoping Sarah would give us a few more examples. One, warfarin, clopidogrel, even statin treatments. But we need that data on clinical utility, and we need to invest in it so that we can figure out the balance of benefits and harms. And one promising area, you've heard about BRCA, but that applies to only the percentages of men and women who have BRCA mutation. How about the rest of us? And the concept of genetic risk scores is gaining momentum right now. This is data from now a few years back based on 80 SNPs or 80 genetic, there's probably 200 or so genetic variants for breast cancer. Individually, they're very small effect size. You put them together, you have the polygenic curve. And this is data I want to take some time to show you that they plotted the 10-year absolute risk of developing breast cancer for women with and without family history by the polygenic risk score percentile. And notice there is this, go back. I guess first thing you notice is that people with family history, regardless of the genetic risk score, are at higher risk. That means two things. There's environmental determinants or missing heritability, or both. But notice that people at the higher genetic risk score, let's say more than the 80th percentile, arrive at a certain threshold. Let's say if you set a certain threshold for women to start mammography, and there is a big debate whether you should have it between 40 and 50. If you have a higher genetic risk score, plus or minus a family history, you might arrive at that threshold earlier. So the whole idea of using genetic risk scores that affects everybody, and for every disease in the population, you have a genetic risk score. And GWAS and other studies are establishing the polygenic contribution to risk. Maybe we can discuss that later. Now, I wanted to give you the tier 3, and obviously we have a speaker today that will talk about Ancestry, but the direct-to-consumer market has gone really way up. And as a public health professional, I've always warned people to think before they spit. And I still do that. I welcome the debate because spitting is good, but on the other hand, you have to know what you're getting. And right now, I don't know what we're getting. Some people think they're getting stuff. Maybe we're getting stuff on the Ancestry side, but not on the health side, and that's a subject for debate. So I want to end up with the 4th theme. So all of this now, all the previous themes are ways to get public health and healthcare to work together to shape the scientific discoveries in the genomic space. But if we do just that, precision medicine will only still have a small effect on population health. So I want to introduce the idea of precision public health. And this paper we published a couple of years ago called Precision Public Health for the Era of Precision Medicine. So what is precision public health? People ask me all that question. So if you're working at the individual level, precision medicine is the right intervention for the right person in front of you at the right time. Some of us work at the population level, thinking about delivering the right intervention to the right population at the right time. That's precision public health. Now, what does public health do? If you ask me, we do 3 core functions. Assessment, meaning figuring out who has what in the population through surveillance and epidemiologic studies. We do policy development, and then we do assurance, meaning trying to connect people to the right services or the right healthcare. So the question is, can we do all these functions with more precision? That's a hypothesis. And the hypothesis is guided by all these data, the big data, whether it's geographic information systems, whether molecular tools, and I'll show you some examples. At the same time I was publishing my paper, the Gates Foundation CEO, Sue Desmond-Hellmann, had essentially arrived at the same idea because she works for, on public health issues. And they published the paper in Nature at the end of 2016, in which they showed some examples. They're working on child mortality in Africa, some of their projects. And the idea of targeting interventions for the maximum benefits of a subpopulation is at the heart of precision public health. So they gave some examples about, does that mean I'm in trouble or what? So there are 3 examples from their paper about mosquito control and then targeting interventions in, for example, in the population by spraying certain areas. So these tools of data science that can help us develop the right interventions is what we're talking about for precision public health. And they do involve molecular tools. And let me show you how. So people know about the C registries. C registries have been around for many years. And they give us estimates of incidence and mortality of different cancers. But cancer is not a homogeneous group. Now, molecular tools, in this case, breast cancer, and this is data from their 2015 data, and this was even before the genomic era. You can show incidence rates by the types of biomarkers, in this case, triple negative, HER2 mutations, HR positive or negative. Imagine what the new tools of genomics will give us in terms of tracking the epidemiology and outcomes of different subsets of breast cancer, maybe the oncotype DX positive or negative. And so the idea of integrating these molecular tools and population activities is still not happening at the large scale. But in order to figure out how to solve the problems of breast cancer at the population level, we need to integrate these tools in the surveillance functions. This is a more CDC-oriented tool. We haven't talked about pathogen genomics. Pathogen genomics tools, i.e. whole genome sequencing of bugs, is now a tool for precision public health. Let me give you an example. The CDC started the Advanced Molecular Detection Initiative, or AMD, a few years ago. And in the first 3 years of the program, they were tracking Listeria outbreaks before and after implementation of whole genome sequencing. And so you have 3 bars in 3 successive years. So what has happened, if you look at the green bar over time, I mean, Listeria is a major cause of foodborne outbreaks, and it's a major contributor to morbidity in the population. But with the introduction of whole genome sequencing compared to the previous assays, now more clusters or epidemics of Listeria have been detected, more clusters have been detected sooner, more outbreaks solved, the median number of cases per clusters have gone down, and more importantly, the number of cases linked to a food source have gone up. This is precision public health in action, and it has nothing to do with the person in front of you. That's not personalized medicine. It's precision public health, and it uses the same tools of genomics and other tools that we have. So in closing, what has happened to medicine over time is that it's evolved from medicine to genomic medicine, and now we have this new buzzword of precision medicine. And medicine and public health have always worked together to solve problems. And as medicine became more genomic, public health had to adapt to pick up the tools from medicine that could be used at the population level. I think now we're entering a new era where next generation medicine and public health will be able to use some of the tools and technologies to do a better job both at the individual and the population level. So with that, I'm going to stop and see if you have any comments. Thank you.
I know we all probably have questions we want to ask right now, but we're going to wait until the panel portion to ask questions and make comments. So after that great talk, I'm very excited to introduce our next speaker, Dr. Sarah Vander, who is an assistant professor of pediatrics at the Vanderbilt University School of Medicine. As a board-certified pediatrician and a medical geneticist, her research focuses on the development of tools to use large data sources such as electronic medical records and DNA sequences to predict and improve children's responses to medication, also known as personalized pediatrics. Dr. Vander has published over 50 research publications, and most recently today in Science, a paper came out with her and her colleagues, as well as a really influential paper in JAMA, Circulation Genetics and PLOS ONE. In 2013, she won the Faculty Development Award in Clinical Pharmacology from the Pharma Foundation. She was also the recipient of a 5-year Burroughs Wellcome Fund Innovation and Regulatory Science Award to better understand children's responses to medications, and we're thrilled to have you here, Dr. Vander.
While he's pulling up the slides, I'll start by just saying thank you very much for the invitation to be here. It's certainly a wonderful place to visit. I'm happy to be here. I'm a little bit not happy to follow Maureen. He gave us such a wonderful, like you said, 30,000 view up of this field and the intersection of precision medicine and public health. I am a general pediatrician. I spend 1 day a week in the clinic and the other 4 or 5 or 6 or 7 days of the week doing research in this area. And my goal today is to get a little bit deeper down into the weeds and talk about some specific applications of precision medicine to give us some more topics to talk about as we move forward in this discussion. So I'm going to start today by talking a little bit about pharmacogenetics. And again, you've heard a little bit about this already from Maureen, and so I'll dive right in. This is not the beginning of my slides. Let me go back to the start. Can you just take me up to the first slide?
Sure. Sorry, I thought you were.
Spoiler alert. Now, everybody close your eyes. Don't watch. Don't look behind the curtain. No, it's okay. There we go. All right. Disclosures, just so you make sure. I don't think you'll see any evidence of that impact of those on my topics. So pharmacogenetics, what does this really mean and how did we get there? Let's start out with secondary prevention in a heart attack or myocardial infarction. I am not a cardiologist, maybe some of you are, and if all of us though can recognize that this is a blood vessel. Okay, it's got contrast in it, and apparently from watching the news this morning, it looks like this blood vessel looks like all of the freeways in Salt Lake City this morning, okay? So traffic was flowing, and then something happened, and traffic is not flowing down here, okay? When that happens on the roads, it's a major inconvenience. When it happens in your heart, it can be a major health problem, a major health incident. Luckily for this particular patient, this is the before image. This is the after image. So an interventional cardiologist was able to go in, place a stent, say open up the roadway, and now those oxygen-carrying red blood cells can safely get to that area of the heart tissue. So this is a save for this patient. When this patient goes home from the hospital, which amazingly will happen in like 24 to 48 hours, I can't believe how quickly people are able to recover from these interventional procedures that the cardiologists do. But when they go home in a day or two, they typically will go home with antiplatelet drug. Very commonly, Clopidogrel is one of the drugs of choice. How did we get there? Why do we give everybody Clopidogrel? Well, the reason is because we have some great population-based data that shows that the endpoints, bad outcomes, things like dying or having another heart attack, are more common in people that are sent home on placebo than people who are sent home on Clopidogrel. So this is a great study that showed us, yes, on a population basis, it would be a good thing for everybody to get Clopidogrel to help reduce the number of people who die within 30 days of their heart attack. So this is not a long-term outcome. This is a short-term save, again, for these patients. But as it turns out, not everybody gets that benefit from Clopidogrel. So a study that came out about 5 years later, and this is one of several studies that showed this effect, showed that there's a genetic variant that people with a TT genotype or high-risk genotype, they look an awful lot like that placebo curve on the prior graph. They look a lot like people who don't get any medication or any benefit from this medication, whereas people with the other genotypes at the same site in the genome, so amongst all of those billions of base pairs, this one little misspelling shifts you from one risk curve to another risk curve. If you have the more common genotype, you benefit from the drug. Thus, we were able to see an effect, a benefit in the randomized trial. But for a minority of patients, it's not going to do them a lick of good. And perhaps they should use a different drug instead that will help prevent them from having these serious outcomes. As a result of these data and other data that showed similar effects, the Clopidogrel label was revised in order to let clinicians know in the form of a black box warning, which clinicians take pretty seriously, that there is this effect. The gene of interest in this case is CYP2C19. It's an enzyme that helps turn Clopidogrel in the chemical form that is in the pill to one that's actually active in the body. If your CYP2C19 doesn't work, that drug cannot be turned into its active form, and it, again, is just like taking that placebo drug. You'll notice, though, if you read the label closely, it doesn't say clinicians must test for this. It doesn't say that you must change your prescription based on the test. What it says is, there's some stuff here that you might want to be aware of. Please be smart and do the right thing for your patient. That leaves it pretty ambiguous, although lots of us physicians, especially pediatricians, we don't like to be told what to do. We like to think about our patients and provide that personal or precision care as best we can. But people looked at the data, people looked at the labels, and in many centers across the country, we did develop programs to enable clinicians to be able to test for that variant in the gene and provide the kind of gene-based prescription advice for their patients. So at Vanderbilt, we have the best acronym, I think, in the whole world for our program. It's the Pharmacogenomic Resource for Enhanced Decisions in Care and Treatment, or PREDICT. We didn't even have to use like the second letter of any of the words. It's fantastic. So that's the name of our clinical genotyping test that's available to clinicians. It includes CYP2C19 testing as well as several other drug-gene interactions, providing that information to clinicians so that they can decide whether another therapy is best for their patient. So this is a picture that spread like wildfire in the Vanderbilt community in October of 2010, which is right when we very first initiated this clinical program. This is a picture of John McPherson, one of our physicians, and his patient, Sybil Van Cleave, who is one of the very first patients whose DNA was put into the assay for this PREDICT program. She had had a heart attack. She got the test. It turned out that she did have one of those high-risk variants in CYP2C19, and upon getting this news and being told that she might need to get a different medication than the usual, her response was, well, I'm not surprised. There's nothing normal about me. And she was very happy to be on our newspaper articles and our ads for the program and all this. She's a wonderful champion for pharmacogenomic testing. She's that N of 1 case where we think we're able to shift her health curve by providing her an alternative therapy. But how rare are patients like Sybil Van Cleave? How common is it that we're going to find somebody that has a variant that can really be acted upon? This is a graph of based on about 10,000 patients that were genotyped at Vanderbilt as part of this program. You can see the different drugs and their relevant genes are listed beneath the bars. Shown in red and blue are the proportion of individuals that had either a very high-risk genotype in red or at least one actionable variant where we would encourage the physician to consider a change in medication therapy for that patient. And as you can see, depending on the drug and the gene, there are variable different numbers of people who are affected by these risk variants. Now, for those of you who are mathematically inclined mathematically inclined in the audience, you will not be surprised at all by what you're going to see on the next click. But I was impressed by how many people are taken aback by this last piece of the graph, and that is this, the cumulative number of actionable drug genome interactions across these 10,000 patients. So in all, over 90% of people tested will have at least one actionable pharmacogenomic variant. So in this room, there are 9% of us, and I've had the test. It turns out that I'm one of the boring 9% who don't have any actionable variants. I have to take the usual doses of all the normal drugs, unlike Sybil Van Cleave. But I am also in that very special 9%, a minority, who have all the boring variants. But most of us have one or more variants that probably should be considered when we are given drugs like warfarin, simvastatin, clopidogrel, etc. So, it seems great, right? How could this possibly go wrong? It's simple. We just test a few variants, provide the clinicians with information, and we're going to make everybody's health better. It goes without saying, right? Well, maybe not so much. So there are lots of caveats to applying this type of testing in giant populations, and I'm going to share just a couple of them. So one issue is that there are a lot of different populations, right? We've heard about minority populations, we've heard about urban and rural populations already this morning. My population that I'm passionate about is children. Okay, so can we use pharmacogenomic information in children? It turns out the answer is maybe. We looked at 42 different drugs that had excellent pharmacogenomic information available in the literature. They're supported by multiple guidelines. We took the 10 of those drugs that are used most frequently in children. Over 500 children per year are placed on these drugs in the Vanderbilt institution alone. And then we looked at how many manuscripts reported the Pharmacogenomic Association in children. Anybody under 18. Didn't even have to have all children in the manuscript. If you had 2 kids that were 18 and a half years old, we took it, okay? We included it in these numbers, and it is sad, okay? There are 2 drugs, risperidone and codeine, for which there are over a dozen manuscripts reporting the interactions of those drugs with genes. And by the way, for risperidone, those papers are conflicting, so we still don't know what to do with that. Codeine now has a contraindication for children under 12 from the FDA because of several issues with the safety of that drug. For the rest of these commonly used drugs where we know that there's a drug-gene interaction in adults, you can count on your hand the number of papers that have explored that pharmacogenomic interaction in children. This is just one example of a special population. We know that in different ethnic and racial groups with different ancestries, there are different variants that drive the interactions between a gene and a drug. And for most of those minority populations, we just haven't done the work to figure out if the same variants that have been seen in thousands upon thousands of European ancestry people are the right variants to help us make the right decisions for those patients. So, another example, another drug, warfarin, is one that we have very well-established interactions between genes and drugs. There are multiple variants in three genes in particular that influence what dose of warfarin you might need in order to be adequately anticoagulated. There was a paper that came out in the New England Journal in 2013 that did a randomized trial of genotype-guided dosing of this drug. And what they found is that with the genotype-guided dosing, the patients that had the genotypes done and the physicians used the algorithm that included genotypes in predicting the dose, they had the right dose of the drug more often, they had fewer INRs over 4, that means that they weren't overdosed on their drug, and they reached that therapeutic level of the drug in a shorter amount of time. So we all pat ourselves on the back and said, yes, we're doing a great job. Again, we can improve population health through this approach. You flip a few pages over in the New England Journal, though, and there's another test, or another report, another randomized trial, slightly different strategy, slightly different population, slightly different settings, and no difference was found between the genotype-guided approach and a non-genotype-guided approach. So here we have conflicting evidence as to whether this really provides a clinical benefit for people or not. Now, it turns out there are a lot of differences between these two studies. One of the major differences, though, between either of these studies in real life is that once people are in a trial for warfarin, study coordinators will be careful to make sure that everybody knows that they need to get their INR checked, or they need to see how they're doing on their warfarin every single day. Okay? And it turns out that the genotype might help you on day 1 or day 2, but once you're checking drug levels, then you know exactly how that person's doing on the drug, and you're going to adjust based on that drug level. The place in the world where I think genotyping will probably help most are places like my hometown of Winona, Minnesota, which, although it's 40 minutes away from the Mayo Clinic, has a somewhat struggling rural health system, community health hospital. People get started on warfarin and they come back a week later and get their INR checked. Okay? There's a place where getting the dose closer to the right level on that first day of therapy is going to make a huge difference, because you're not checking it the next day and readjusting for 7 days in a row. I think that's where we have the potential to make an impact. That's also probably the last place that will get a randomized trial funded because of the constraints in making that happen. So this is one of the catch-22s that we find ourselves in when it comes to precision medicine. Another caveat, another difficulty, another issue that we think about a lot when we think about genomic medicine is incidental findings. So when you read the genome, you're going to find variants that you didn't plan on. We did a study several years ago, and it was very focused. Okay, we're not talking about, or for this next couple of slides, I'm not talking about whole genome sequencing or whole exome sequencing. This was a very focused study where we looked at just two genes that are associated with arrhythmias in adults, and we looked at about 2,000 individuals. So we sequenced the entire gene, and when we did that, we came up with lots of variants that we hadn't seen before, and we didn't know what to do with. Were they really an incidental finding, or were they just, an accidental misspelling in the genome that nobody really cares about? Because we know that there are lots of those biologically found as well. So what we did with all the variants is we sent them to expert laboratories, places that are highly familiar with these genes, who have for years been interpreting these exact kind of results. And what did we find? So remember, we had 2,000 people. We found 122 non-synonymous and splice site variants. So the kind of variants that we think that we probably should be worried about in these genes that can cause life-threatening arrhythmias. And these were found in 223 individuals. So right away you're thinking, okay, something's very wrong, because 10% of the population doesn't drop dead of arrhythmia. Because I know more than 10 people and they haven't done that. So there must be something. These can't all be pathogenic variants. So we took the 42 variants that were relatively rare, because a couple of those variants of the 122 were very common. You know, they're found in large numbers of individuals in our own population as well as in published populations. So we're like, those can't be it. Let's take the 42 variants that are pretty rare, because those are the ones that we're most worried about, and let's send them to labs and see what they say. And the labs came back. We thought they would be in agreement. This is the Venn diagram of what 3 different expert labs thought about the exact same data. Wow, what are we going to do? We have to come up with better ways to figure out which variants we think are going to really impact health and which variants we think are nothing burgers. So that takes me to the next little piece of my talk, because it ties into this idea of incidental findings. And that is a new study. This is the one that just came out in Science this week, talking about phenotype risk scores. Many of you are likely familiar with the concept of genome-wide association. For those of you who aren't, it's not rocket science. What you do is you take a phenotype, a trait, an outcome, something that you can see or observe or measure in a human being. And instead of looking at one gene at a time, you take 1 million variants or 10 million variants, or if you're a pediatrician on a budget, maybe 500,000 variants. And you do a statistical test across all of those different variants to figure out which of the variants are associated with your outcome. Okay? And the way we plot these is called a Manhattan plot. It's not from Dr. Manhattan or invented in Manhattan, but it's called that because you hope to see something that looks like the skyline of Manhattan. So this is the negative log base 10 of p-values. I don't work a lot in logs in my regular life, but what you need to know is that the taller the peak of the skyscraper, the more statistically significant that finding is. And then all of these little dots, and they're all crunched together in the bottom here, each represent a place in the genome. They're just mapped out by location. So again, this is an example. We see a significant finding here. Anything above the red line we count as statistically significant. That's GWAS. And it's been enormously helpful. Like most new technologies, it was probably oversold in the beginning, applied to a bunch of places where it maybe wasn't as helpful, but in the end, it has its uses. Several years after genome-wide association was invented, phenome-wide association came along. And this is the exact same idea, the exact same premise, but it's turned around backwards. So here we start with a genotype. And instead of looking across a genome, we're going to look across the phenome. Well, what is the phenome? It could be any number of things, but one of the most common ways so far to use this is to look across billing codes. Because whenever a physician diagnoses a patient with a condition, we have to associate a billing code with that. And there's a whole industry around making sure those billing codes adequately represent what the disease process is in that individual. So you can group those billing codes into what's called Phe codes or PheWAS codes, and it gives you a picture of what diagnoses are associated amongst patients with particular genotypes. So again, you see an example of a Manhattan plot. This one doesn't have the filling in of the skyscrapers. It looks, without the dots on top, sometimes people call it a Manhattan, Kansas plot, which is not what you hope for. So, through the years, we've discovered and come up with ideas, and again, you heard a little bit about this already this morning, that we shouldn't just look at the individual dots that pop up on the top of our GWAS plots, but we should probably consider the effects of these things as they can be added together, as they can be combined. And this gives us polygenic risk scores that help us look across multiple variants and multiple genes. And Lisa Bastrach, Josh Jenny, and others at Vanderbilt, and a team that I was able to be a part of, took that same premise and applied it to PheWAS and developed something called phenotype risk scores, where instead of looking at one particular phenotype, you look at the different features that could go together in order to define a disease. It turns out that this is a relatively simple mathematical formula that was used. You could imagine that if runny nose was one of the phenotypes on this list, that probably wouldn't be very informative, because at least in my clinic, everybody on the planet gets a runny nose at some point. But something that's very specific for a disease, like a pseudomonal pneumonia that really happens in a select group of people, and most commonly in cystic fibrosis, could be something that could be highly informative. So what Lisa and myself and our team did is weighted the phenotypes, weighted the things so that the most common things only contributed a little, and those more rare phenotypes contributed a lot to the phenotype risk score. So started out with 6 different diseases, assigned the phenotype risk scores based on these phe codes and what we knew about the disease. And you can see here, these are the differences in phenotype risk scores for people who had already been diagnosed. This is proof of principle. People who had already been diagnosed with the disease versus those who did not have the disease, based on electronic health records at Vanderbilt. So it worked excellently for cystic fibrosis. We see a huge difference in the phenotype risk score for those with or without disease. And by the way, the code for cystic fibrosis is not included in the phenotype risk score. We strip out the disease itself out of the mix. It worked for Marfan, achondroplasia, hemochromatosis, Li-Fraumeni, and then allegedly the wheels came off the bus with PKU. Well, why is that? Well, that's because we do newborn screening for PKU. The features of the disease are not present because we intervene as pediatricians, as geneticists, and make sure that the cases and controls never get the actual phenotypes that we would search for in the FRS. So this provided a nice proof of principle that we're actually doing what we think we could do. So Lisa and Lisa is a data scientist. She's not interested in looking at 6 diseases. Whenever she sees something interesting in 6 diseases, she says, how can I apply this to all diseases? That's just the bioinformatics style. So what they did is they leveraged the fact that in OMIM, there are almost 4,000 diseases that have already been mapped to these things called HPO terms. So there are about 6,000 HPO terms, and each disease in OMIM has a list of which features, based on their HPO terms, are present in patients with that disease. The only problem is HPO terms don't live anywhere really outside of there. As a clinician, I don't write down which HPO terms my patient has, but we do have the phe codes. So what Lisa and her team did is they built a map of all of the phe codes to as many of these HPO terms, linking them together in as many times as possible. So I like to think of it as they made the Rosetta Stone between the phe codes and the HPO terms in order to enable us to look in electronic health records for all of the Mendelian diseases. So what can we do now that we have this tool? Well, I'm going to spell out just 2 applications, but I think there are probably more. I said these data are brand new, so we'll be coming up with new things as we talk to people such as yourself. So one application is understanding variants. As I ended the pharmacogenomics piece, I mentioned that there are all these variants that we're not sure if they're pathogenic or not. It turns out we have lots of those patients in our BioView biobank at Vanderbilt. There are 21,000 some patients who have been genotyped as part of a exome chip genotyping platform, and in those 21,000 patients, there were over 6,000 rare variants that we weren't sure if they were pathogenic or not. They've been unassigned in the literature to date. So picking just one of those for the sake of example, there's a variant called R76S in the SUOX gene. Nobody knows what this does. We know that SUOX is associated with a disease called sulfocistinuria, but we don't know if this particular variant is associated with that disease or not. So we looked. We took all the patients that had that variant. It turned out that there were 21 carriers of that variant. We calculated their phenotype risk score for the specific disease of sulfocistinuria. We calculated the same thing for everybody who didn't have any variants in this gene, and it turns out there's a huge difference. The p-value is 1 × 10⁻⁶ for the difference in the phenotype risk score between these 2 groups. We thought that was pretty good evidence that there's something biologically different about people who have this variant versus those who don't. In other words, this looks like it's a pathogenic variant. We did biochemical assays to show that indeed the R76S variant impairs splicing of this gene, which is exactly how known variants that cause disease in this disease work. So we're able to move the needle from this being an uncertain variant to one that we believe is pathogenic. The other really interesting thing about this finding is that until today, sulfocistinuria has been defined as an autosomal recessive disease, meaning that you're supposed to have to get 2 damaged copies of the genes in order to express this disease. These 21 carriers in whom the phenotype risk score was floridly different than everybody else were carriers. They only had 1 variant in the gene. We did sequence the gene for all of these individuals to see if there were other variants present that could be defining maybe they're a compound heterozygote, and they were not. We don't see any other reason why these people have traits that go along with sulfonylurea. Do they have the full-blown disease? We don't think so. It doesn't, they're functioning, they haven't been diagnosed with this disease, so they appear to have a mild form of the disease. They have some of the features, but they're not fully expressing the disease. So something that we thought required 2 hits to your genome, it turns out is probably more of a continuum of disease, and I think this is a theme that we're going to see a lot as we learn more about our genomes. Application number 2, and some of you may have thought of this already, is that now we have a tool by which we can search for individuals that have these diseases. This is a graph of the accumulation of traits that go along with disease over time. So as a patient is seen by more and more doctors, they get more and more diagnoses. If these particular phe codes are the ones that contribute to phenotype risk score, their score will go up over time. In this particular example, you can see you have a sort of low risk, medium, higher risk, and wow, it's really elevated here. This red line represents when this person was diagnosed with their genetic disease. So our thought is that we could perhaps let people know, let clinicians know earlier that they have a patient. Have you thought about this particular diagnosis? Can you put the pieces together? Can we help make clinicians connect with the various subspecialists to make sure that everyone's thinking about how to best diagnose these patients? And this is one of the applications that we're really fired up about at Vanderbilt. This is an image from our Vanderbilt newspaper that came out today. This is Lisa Bastrach, the super smart data scientist who really led all of this work, and Josh Denny, who's our head of precision medicine at Vanderbilt. And it's just been a fantastic ride. I think this is a technology that's going to help us enormously in the years to come. And with that, I'm going to transition to the last little bit of my talk, and that is to switch gears. We just went from pharmacogenomics to something that, finding Mendelian diseases, but that if we use the phenotype risk score, you could imagine that you don't even need any DNA, right? We could potentially diagnose genetic diseases with no genes, kind of the way that geneticists have done it for years and years, where they look at the patient, they see the features, and they make the diagnosis with or without the DNA to confirm that diagnosis. So my point being that precision medicine isn't just about genomics, and we need it to be not just about genomics, because that's only a small part of what contributes to the person that each of us is, and to the disease processes, to the health disparities, to the health changes that we see in our patients. So I'm going to show an example of some work that I led at Vanderbilt looking at acute kidney injury risk prediction that has nothing to do with genomics, but everything to do, I think, with precision medicine. For those of you not familiar with acute kidney injury, it's defined as a quick change in your creatinine level, which is a lab test that measures how well your kidneys are functioning. Unfortunately, this is a problem that occurs in a large minority of patients, pediatric patients, who are admitted to the hospital for other reasons. And it's because a lot of the medications that we give, a lot of the interventions that we make, and a lot of the diseases that cause them to come into the hospital can be hard on their kidneys. Unfortunately, also, it's associated with increased morbidity, increased mortality, and increased length of stay. So, like I said, this is a different kind of precision medicine. Using that same sort of thinking, though, as the phenotype risk score, we wanted to do surveillance of the electronic health record, looking for things that would help us know whether or not a child was at risk for acute kidney injury, such that we could encourage clinicians to test for this by ordering a serum creatinine. And you might say, like, well, why do you even have to test for it? The reason is that the vast majority of pediatric patients come and go from the hospital without even getting this lab draw tested. I know in adults, there's oftentimes the habit of getting daily labs in patients, but in children, we try to keep all of their blood inside them because they just don't have as much. So we need to know whether or not a child is at risk, so we know whether or not to test to determine if their kidneys are fully functioning or not. So basically, we took as much information we could that's available in electronic health records. We culled through all of those data to find the best predictors of acute kidney injury in those patients that we knew that had it, and using those that we knew didn't have it as controls. 20 years ago, we might have made something like this. This is a nomogram that incorporates all of the risk factors that we found. And we could print these out, maybe make a nice badge card. People could carry around rulers and calculate out how much kidney injury risk each patient has. But it turns out it's 2017 at the time that we were rolling this out. You don't need to have slide rules and pocket cards. We have electronic health records, right? We can use incredibly complex calculations using data that's already in electronic health records, the stuff that we're already gathering as part of routine clinical care. The computer can do all of these calculations for us because computers are super good at that stuff, and just let us know what the risk is for each patient for each moment in time. So for example, this is a graph. Each different color is a different patient that was admitted to our pediatric hospital, and you can see their risk of acute kidney injury as calculated over time, in real time, as they work through their stay. We took this information and we said, you know what, if people children are at a 50% risk or higher of acute kidney injury, let's let their team know, and let's ask them to check and see if their creatinine is normal or not. Because if it's normal, they're good to go. If it's not, then we need to be much more careful about how we're taking care of that patient. Now, again, what could go wrong, right? We know that this, we did a lot of work to validate our model. We know that this is a problem in pediatrics, but we have no idea whether this intervention would be helpful or not. So rather than just deploying this across the Vanderbilt health record, or even maybe asking other centers to do the same, we decided to embark on a randomized trial to see if our intervention would make any difference or not. So we built actually two statistical models, one in the ICU and one for the non-ICU patients. And then we randomized every patient that was admitted to our hospital over the course of a year. And you can see that it's about half and half patients that were randomized to receive that information, where their clinicians were alerted if they exceeded the risk for acute kidney injury, or if they were not at risk. They didn't see the prompt. In the control group, nobody saw that alert. We then took a look. We could see which patients were at risk because we actually calculated risk for everybody. And you can see the risk was well balanced across our different groups. But what we wanted to compare is, within these people at risk, is there a difference in how much screening there was for serum creatinines as a first look at the process outcome of if we're making a difference with what we're doing. And in the ICU population, we did see a statistically significant difference, right? In the control group, we had 59% of patients actually got a serum creatinine tested, whereas in the intervention group, we had 68%. Absolutely not perfect, absolutely less impact than we thought we would have. It turns out clinicians don't care what I think the patient's risk is, apart from a few of them. So we made a difference, but not nearly the size of difference that we think that we need to in order to make a dent in this disease. And then in patients on the floor, there was no statistical difference. My colleagues who take care of patients who are not critically ill really don't care if I let them know that the patient's at risk for acute kidney injury or not. They paid no attention to our intervention. So why do I show these data? Because there is a paucity of these kinds of randomized trials for any kind of intervention that we make in pediatrics, in adult medicine, in academic health, and community health. We need to test to see whether we're doing makes a difference or not, because those interventions where we don't make a difference are just as important for the whole world to know about than the ones that we do. Otherwise, we'll just be spinning our wheels and spending resources on things that don't make a difference in health. So, normally at the end of a talk, I like to give conclusions, but it is very early in our day, and I think this is a discussion that's going to continue in the hours to come. So instead of talking about conclusions, I'll just kind of remind you of the themes that came up really over the past hour or so. And that is, I think there is potential for health benefits from the application of precision medicine. I think the naysayers have some good points, but I think we will prevail and make a difference using these approaches. We can improve medication doses, we can improve medication choices, we can enable targeted screening of individuals, whether that be for genetic diseases or non-genetic diseases, and hopefully we can move towards earlier and earlier detection and treatment of disease. But there are definitely ways that we can miss the mark. And again, I hope you've picked up on that. One way is we fail to measure, fail to include the diverse individuals that are going to be consumers of these kinds of approaches. There are definitely unintended consequences whenever we have a new technology, a new approach, and we need to test our interventions. If we fail to test them, we won't know what works and what doesn't, and we will likely drown in the noise. So with that, I'll wrap up, hand things back over for our panel.
I don't know about you, but I learned a lot of good stuff in that session. We're going to take about 20 minutes for questions and discussion. If anybody do we have microphones? We have microphones, so Bridget is going to go around with the microphone. So whoever wants to ask the first question, go ahead, and Bridget will bring you a microphone. Because we're recording this, so if you could just wait the 2 seconds to get the microphone, that would be great.
I'm Aaron Myers in the Department of Medicine. Those were terrific presentations. Thank you very much. I'd like to comment and follow up the closing theme summary. All of the data, the favorable data that both of you reported, had to do with relative effects. In other words, an intervention produced improvement. That's like driving to Chicago and passing Evanston, Wyoming, just a few miles to the east. We're moving in the right direction, but if your goal is to get to Chicago, there's no reason for celebrating when you get to Evanston. You've got a long way to go. So the goal, I believe, that we all have is to make the right decision the first time and every time for every patient. And I want to point out that providing reminders for clinicians, giving me a risk for a phenotype, is helpful but not very effective. Since Lister introduced antisepsis at the end of the 19th century, we've known about hand washing, but all of the data indicate that isn't carried out very well in most clinical institutions. In fact, some of the data I use when I present this comes from neonatal ICUs, the most vulnerable population you can imagine, premature babies with IV lines who are being evaluated for sepsis, and even though 40% of the nurses don't wash their hands and do proper protection. So my question is, have you thought about using electronic rule-based systems to provide not reminders, but instructions and even automatic generation of care? For example, mechanical ventilation of preemies or babies or adults could in fact be done automatically and eliminate people like me from the loop and eliminate the variation we introduced.
I think that's a great reminder. I'll push back a little on the celebration in that I celebrate whenever I can. So, and if you're driving from here to Chicago, you definitely need to celebrate along the way, because that's a long way, and you have to go through, I think, Nebraska, which is hard. But more seriously, I think you bring up a very valid point that the goal is not to just be better, right? The goal is to help our patients be at their very best. With the idea of, can we take some decisions out of the hands of clinicians to eliminate variability? Maybe. And I'll tell you one of the very practical things that we've run up against at Vanderbilt, because we love building our decision support, we love automating things as much as possible. If we put a test if we create an order for a test that the computer orders the test, then we don't get paid for that test. A physician or other healthcare provider has to be the one that makes the decision to choose the test, or we don't get reimbursed. Now, should that paradigm change? Probably, because I think hopefully we'll be able to do the work to prove to people like insurers and payers that there are some interventions that just need to be done, and that's going to improve care and save money in the long term. But for right now, there's that practical barrier. So number one, paying. Number two, evidence. And number three, I think that there's always more things to consider. Now, the hand washing example, you're absolutely right. Outside of an infant having an acute event, that precludes the first provider from washing their hands. Everybody should be washing their hands all the time. That's what it is. For things like pharmacogenomics, it's usually not just the variant that decides what the best medication is for the patient. So right now, I think we rely on human beings to weigh the evidence, because usually the right answer for each patient, for example, if the alternative medication to Clopidogrel is something that they are absolutely not going to afford and take exactly 0 pills of, that's not going to do any good. So there needs to be some, at this stage in the game, some human beings taking in all of the information and making the best decision possible. That's not the end game, but that's where we are today. So you're right, we're maybe only an hour or two east of here. Marty?
Thank you.
Great. So my question is for Dr. Van Priest and others in the audience with respect to the interpretation of the warfarin and the role of genotyping in warfarin. So if you just look on the surface, you would say, well, I'm not going to genotype anyone because the European study showed effectiveness, the US study didn't. What are we to do? Just throw up your hands. But I think if you look a little bit deeper, you would possibly consider that the European study worked because Europeans are Caucasians, and Caucasian data with respect to genotype is actually fairly strong. And in the US, the study, I assume, was a little bit more diverse. And so I think then perhaps the message is we need to do a better job of genotyping non-Caucasians, African Americans, Asians, whose genotype relationship to warfarin dosing isn't quite clear. Make that better, and then maybe everyone would benefit by the genotyping.
Yeah, absolutely. I think you hit the nail on the head that the subset analysis of the African American patients that were included in the trial actually did worse with the genotype guided therapy, and I think that's because we have not included the correct variants for that population. And there are a lot of reasons to include diverse populations in our research studies. Number one, it's the right thing to do for those patients. Number two, you can take a completely selfish approach and say, you know what, it's any variant that we find in that population is also going to be present at different rates in other populations. So it serves everyone's needs in order to include everybody to improve the prediction models. But I absolutely agree. And
Sure. Good morning, Josh Schiffman. Great talk from both of you. What a great way to begin the day. So my question is about newborn testing, and it's going to involve both of you and maybe the whole room if we're lucky. So we all know that the cost of sequencing is coming down, and I don't think it's going to be very far into the future when everyone is getting their DNA, maybe on a newborn blood spot, completely sequenced. The question, and I would be in favor of that, but the question is, you talked about, finding some BRCA1, BRCA2 genes, some of these adult onset cancer predispositions. How do you foresee it working that you would have this information today, and from a clinical perspective, we don't test early on until the actual genes are going to cause a problem, right? So we wouldn't test a young girl, an infant for BRCA1, BRCA2 intentionally. So what are we going to do with this information? We're going to hold it in an envelope until you reach of age, and then suddenly open it up and say, okay, you're at risk? Because there are very real dangers of knowing that information ahead of time. And then the converse is true, right? Sarah gave a great example in pediatrics of it'd be very important to know some of these pharmacogenomics genomic information. You can imagine if we had that for everyone, it goes right in the medical record, that would really change the way that we do precision public health. So just open-ended, how do we actually implement this? What do we need to be aware of?
Okay, great question, and was looking at Jeff Botkin in the audience there, who probably has a better answer than me, but I'll give it a shot. So let's go back and look at the purpose of newborn screening. Why is it a public health program? The whole idea behind newborn screening is that you have, in a sense, some kind of a public health emergency that you have to intervene very quickly after birth, otherwise you have irreversible damage, maybe death, maybe disabilities. So so newborn screening started as a public health program, and more conditions were added, and now people are exploring whether you get the whole sequence. I think you've answered the question. There is so much unneeded or unnecessary and potentially harmful information that doesn't fit in the kind of rubric of a public health emergency. A BRCA mutation, you don't need to know it at birth. I mean, you have the right appropriate age for testing or counseling first. The other thing is that kids or newborns don't have the ability to consent. I mean, if you're going to do a study, and there are investigations that Genome NHGRI has invested in, which is to look at newborn screening as a potential tool for practice. The way I see it is right now it's too early to intervene in newborns. The sequencing of newborns is fraught with ethical, legal, social implications. Let's focus on where the action is. And I'm glad, and I didn't give you the compliment, Sarah, but we need to get evidence. And you showed us data that goes well beyond just showing associations from records, but actually randomizing, looking at outcomes, whether or not we can get to Chicago with one set of tools. We need to do the studies before we do any implementation. So my thoughts on newborn screening is not yet, and maybe some other people will disagree.
You're very patient.
Question for Dr. Van Cleave.
Wait, just 1 sec. Oh, button on the bottom.
How can we take care of patients when we can't work the microphone?
That's a good point.
Precision microphones.
I have a question for Dr. Van Cleave. So I'm on a committee that's curating genes for platelet problems, and I suspect there are people in the room who are interested in curating a lot of genes for their phenotypes. Sounds like what you described in your science work be a phenomenal tool.
We hope so. We hope it helps lots of folks.
So, do you want people to collaborate with you?
Yeah, absolutely. I think, take a look at the paper. You can reach out to me. You can reach out to Lisa and our team, and we'll figure out the details. If I'm not the right person to work with, then we'll get you connected with the people who are. So, but the goal was to keep it simple. In building the risk scores. There are some, of course, nuances to it and details that there's a reason it took several years to get from the idea stage to the publication stage on this. So our hope is that by spreading the word, we can smooth out that translation time for other centers.
You know, I am so sorry. We are so over time at this point. We could talk about this all day and just forget about all the other people, but I don't think that will go over well. So we would like to really, really thank these great speakers today and all your questions.